You know, Mr. Mendel was quite an intelligent person. While experimenting over pea plants, he discovered 3 laws which are called as Mendel's laws. So in this video, we will learn in detail about these laws. Like, share and subscribe. The 3 laws of Mendel's are Law of Dominance, Law of Segregation, Law of Independent Assortment. First is the Law of Dominance. In a cross of parents that are pure for contrasting traits, only one form of trait will appear in the next generation. All the offsprings will be heterozygous and express only the dominant trait. For example, R, which is for round, and r, which is for wrinkled. So, we will get heterozygous round seeds, R and small r. We can see another example given below with the Y and small y. Capital Y denotes the yellow color and small y denotes green color. So here we get 3 genotypes yield and 2 phenotypes. Second is Law of Segregation. During formation of gametes, eggs or sperm, the 2 alleles are responsible for a trait separate from each other. Alleles for a trait are then recombined at fertilization, producing the genotype for the traits of the offspring. C. Pink color is for tall and light brown color is for short, and the gametes of the parent will be T and small t. After crossing them, you can see the phenotype ratio, which is 3:1. Third is Law of Independent Assortment. Alleles of different traits are distributed to sex cells and offsprings independently of one another. This law can be illustrated by a Dihybrid Cross. Let's take a look to the Dihybrid. Dihybrid Cross is a breeding experiment that tracks the inheritance of 2 traits. It can be any. Mendel's Law of Independent Assortment. So each pair of alleles segregates independently during the gamete formation. For this, there is a proper formula, 2 raised to the power n, where n is the heterozygotes. So below are the given 2 questions, R, small r, Y, small y, and AA, BB, CC, DD. So you have to find out the answer for this. So these are the answer. Because we know that the 2 traits were given in the first part, so 2 raised to the power 2 will be 4 gametes. And 3 traits are given for the second part, so there will be 8 gametes. Now another example for a Dihybrid cross, the traits which are taken its seed shape and a seed color. So the shape is round and wrinkled, R and small r respectively, and seed color is yellow and green, Y and small y. So after crossing, these are the possible gamete combinations. So this is how you have to make a cross, and this is going to be a result. We get the ratio as round yellow 9, round green 3, wrinkled yellow 3, wrinkled green 1. 9:3:3:1 will be the phenotypic ratio. So this has been showed with using the figures. Let's study the incomplete dominance or codominance. So incomplete dominance is when F1 hybrids or the first generation hybrids have an appearance somewhat in between the phenotypes of the parental varieties. So we took the example of the snapdragon flower. Red is for R, and white small r, and this is how we are going to make a cross of it. And these are the results. All are the heterozygous pink, R and small r. That is called incomplete dominance. And now codominance. When 2 alleles are expressed in heterozygous individuals. For example, here we take the example of the blood type. Type A have IA or IAI. Type B is IB or IBI. Type AB is IB, and type O is II. So we have to know what is I here. For that, we will study about the inheritance of the blood group. We know that there are 4 kinds of blood group: A, B, AB and O. And you know that blood group is controlled by 3 alleles, which is IA, IB and IO. I stands for immunoglobulin. IO is recessive. and 2 IO alleles must be present for the person to have type O blood. And IA and IB are codominant, but both are dominant to IO. If a person receives an IA allele and an IB allele, their blood type will be AB, in which characteristics of both A and B antigens are expressed. Because IO is dominated by IA and IB alleles, a person with blood group A could have the genotype IA-IO or IA-IA. This has implications when having children because if both parents carry the IO allele, a child could be born with the genotypes of IO-IO, which is mean the blood group O, even though neither of the parents have this phenotype. So you can see that this is the blood type and this will be the genotype. Let's see the codominance problem. If a homozygous male type B, IB is there, and heterozygous female have type A, IA and I. So after crossing, we will get half of IA-IB and half of IBI. That means 50-50% of both of them. So another codominance problem is if the male have OII, if the male have a blood group O and a female have a blood group AB, okay, then after crossing we'll get the 50-50% the same results. Now, introduction to the sex determination. Sex determination takes place with the help of genes inherited from parents. Particularly, genes are present on chromosomes in the nucleus of the cells. So for sex determination, how the process is done, you can go and watch my short also. I have prepared a short video on that. Male and female gametes contain chromosomes. Male have XY and female have XX. Fertilization of these gametes lead to the formation of zygote, and then zygote is developed into embryo, and further it develops into new individual. The biological process through which sexual characteristics of an individual develops, making it a male individual or a female individual, is called sex determination. Now let's take a brief look over sex determination in humans. So normal humans have 46 chromosomes, that means 23 pair of chromosomes. We know out of these 23, 22 are autosomes, which are same in both male and female, but 23rd pair is heterosomes, which differentiates a male and a female. So we know that male have X and Y and female have XX. In males as well as female, the gamete, that is sperm or ova, contain 23 chromosomes, that is half of 46. More specifically, male or female gametes contain single chromosome. Ova in female contains only X chromosome, whereas in male have sperms contain X chromosomes as well as half of Y chromosomes, which we can see in this following figure. See, human cell is there and 23 chromosomes of male has been described, X and Y, XX and XY, both are 44, okay, but the other 46th chromosome is XX and XY respectively in male and female. This is after cross. If XX is there, then it will be a female child, but if XY is there, there will be a male child. So at the time of fertilization of male and female gamete, if a sperm have X chromosome fused with ova containing X, then the pair is XX, which means female, and XY means male. That is how a sex determination of a child is done. Okay. Environmental sex determination. In loggerhead turtles, sex can be determined on the basis of temperature in which the egg is developing. For example, if the egg is developing at 28 degrees Celsius or below, it produces male turtle. But if the temperature is 30 degrees Celsius and above, then female turtle is produced. So in case of snails having ability to change their sex with respect to the partner. So this is about the two animals, but we are going to study about the details in higher classes. Thank you for watching this video. Please like, share and subscribe and hit the bell icon to get